In this tutorial, I will explain you how to download a gene sequence from NCBI genome browser. And for that, first we have to go to this website. In this website, there are so many things that is related to different fields and research objectives. We will go through all of them step by step. But first of all, we have to open this website, the National Center for Biotechnology Information (NCBI), to find the gene sequence or download gene sequence. So how I get here? You just have to write NCBI, and this will open this link. And just open this link, and from here, let's suppose if we want to download a gene sequence, and for that, let's say we select here gene, and any protein or gene sequence name you remember, like TP53, that is tumor protein TP53. Just search it. When we search this, it will give a lot of different options and outputs. For example, here you can see that there is a different species like Homo sapiens, musculus mouse, a lot of different results. But let's suppose we want to look for the human sequence. We have to click this. After clicking this, it will open the this output. Here you can see that this is the gene ID and TP53 protein, and on this page there are so many information related to TP53, but in this tutorial we are just looking for gene bank ID, that is the DNA database of NCBI. So for that we go there down in this heading, genomic regions, transcripts and products, and here you can see there is one word written FASTA format. So this FASTA format is actually the format which is used to store the ref sequences or annotated gene sequences in a format in all databases. And if you click this FASTA format, you know what is a FASTA format. Here you can see in this figure, that in this page, that the FASTA format actually means that in the beginning there is a greater than sign, and then the description of the sequence, and from the next sequence all the sequence is arranged without any space and in capital letters, and that is called the FASTA sequence. So this is the FASTA sequence of TP53 gene, right? So I think this is very simple and straightforward. There are other things which may come in mind, like download the protein sequence. We will discuss that later.